All right.
Let's welcome the speaker.
Thank you for the introduction. Before I start, I would like to congratulate the organizers for putting this excellent schedule of talks, and it was really nice to meet also people I have read papers about and now to see them in Zoom, and hopefully next time maybe in person. So today I'm going to talk about our computational studies on Cyanobacteriochromes. So the outline of my talk is that first I'm going to mention what are actually these Cyanobacteriochromes, and then I will talk briefly about the methodology for QMMM simulation and how we do this, how we obtain the spectra. And I have two sections of results. So let's start with the introduction. Cyanobacteriochromes are newly discovered photoreceptor proteins that, as the name suggests, are found in cyanobacteria, and they are related to the common superfamily called Phytochromes, which are predominantly found in plants, but also in other organisms. So I guess the next question that arises is, what are Phytochromes? And these are actually large photoreceptor proteins which consist of 3 domains, and usually they come as dimers. So here you see this typical architecture. They have 3 domains. Let me switch to the laser. Okay, so they have these 3 domains called the PAS domain, the GAF domain, and the Phi domain. They are linked together, and they usually form dimers. And this orange-colored GAF domain harbors the chromophore, which is a linear tetrapyral. Now, in contrast to these phytochromes, cyanobacteriochromes are much smaller. They require only the GAF domain, so they are very compact, and on average the size is 20 kilodalton. And similar to the phytochromes, they have the chromophore, which is composed of a linear tetrapyral, so it looks slightly similar to, maybe you are familiar with hemes or chlorophylls or porphyrins. It's actually a metabolite of them. So there are 4 rings, there is no metal in between, and the link between the A ring and the D ring is broken. So that's why they're called linear tetrapyrals. They're linked to the proteins through a cysteine residue, and they form the chromophore. Now, why is cyanobacteriochrome? Why are the cyanobacteriochromes so cool? Well, they absorb a wide range from the near UV to the near infrared. So in contrast to the classical phytochromes, which in terms of spectral tuning are quite boring because they absorb only in the red and far red, the cyanobacteriochromes cover the whole visible spectrum and beyond. So the question arises, how does the protein environment achieve this tuning? Now, in addition to just the spectral tuning, they also have a higher fluorescence quantum yield compared to phytochromes, so they can be used for a variety of biotechnological applications. And here are 2 examples. One is in the optogenetic experiments, and the other one is to be used for super resolution imaging. So we set out to study one representative system, and we studied a cyanobacteriochrome from Cynocystis, which is a genus of a unicellular freshwater cyanobacteria, and it's something like the working horse in photosynthesis. So I guess in the photosynthesis community, this Cynocystis cyanobacteriochrome is something like the hydrogen molecule in quantum chemistry. So it's a very well studied and known model system. And it contains the gene of this protein called SLR1393G3. So it's the 3rd GAF domain, and this is just the name of the gene. This gene encodes a protein of 156 amino acids. And what makes this protein special, why did we select it, is that the resting state, so in dark without light, it has an absorption maximum which peaks at 649 nanometers. And then once this red peak is absorbed, is excited, or red light excites this protein, then it converts to a photoproduct state which has a peak in the green at 536 nanometers. And this is in contrast to all the phytochromes, because the phytochromes, they absorb originally in the red, and once they do the photoconversion, the product state absorbs in the far red. So here the tuning, spectral tuning, meaning the change in the absorption maximum, is going to the red, while in Cyanobacteriochromes it's going in the opposite direction to the green, and the shift is also much larger. So the underlying reaction, how to convert the red form to the green, or also vice versa, green to the red, is by photoisomerization of the D ring. Okay, so of course we are not the first one to study this protein. In fact, there has been already a large body of experimental studies. And in this recent review, Rockwell and coworkers, they have summarized different suggested mechanisms from experiments. So in one case, it was suggested that the chromophore is losing a double bond. I highlighted this bond here. So it's called the isomerization from ficocyanobylene on the left side to ficoviolobylene on the right. The other mechanism was proposing protochromism. Here, the nitrogen of the pyryl ring C is getting deprotonated, so I highlighted it as well. Then the next mechanism was saying that there is a tryptophan residue which is doing pi stacking with the D ring, and upon isomerization, the tryptophan is pushed away by the D ring, and then water can enter the binding pocket and induce a shift to the green. And the last mechanism was proposed to be a trap twist, where the D ring does not fully isomerize; it remains highly twisted, and therefore it affects the conjugation lengths. So in the meanwhile, since 2014, there have been NMR and resonance Raman studies, which have ruled out the first two mechanisms. So there are only two mechanisms remaining, two proposals, and we set out to address it. However, when we looked into literature, there have been actually no studies of cyanobacteriochrome photochemistry. And there are two reasons for this. One is that actually the size of the chromophore, you see here, these are the usual chromophores from the photoactive yellow protein, GFP. This is a regular tryptophan, flavin. We heard already today two talks about all-trans retinal, and this is the chromophore in cyanobacteriochromes. So retinal has 54 atoms. This linear tetrapyrrole has more than double the number of atoms. And the other reason for it is that there is a lack of high-resolution crystal structures. So we want to have a structure in order to do QMMM simulation. So we need a red form and we need a green form. However, they were obtained for different proteins, which kind of doesn't allow you to do a direct comparison. And there was also a blue form from a different protein. Okay, so very recently, the group of Wolfgang Gärtner has solved the crystal structures from the same cyanobacteriochrome for the red form, which is called PR, and the green form, PG. And this was done for exactly the same protein, which is this SLR 1393. So here's the first part of our results, which was obtained by Christian Wiebler, who was postdoc in my group at the time. And so before I go to our simulations, I would like to introduce the methodology, because as far as I understand, there are also some experimentalists in the audience. So there is this important development of the multiscale modeling, which was awarded a Nobel Prize in 2013. And it basically says that similar to the photograph here of Alfred Nobel, which is divided in 3 different zones of different amount of resolution, okay, we have, if you want to recognize the person, we need to focus the highest resolution on the face, because this is the characteristic. And then once we go out from the central zone to the outskirt, the resolution is getting lower. We see that the picture is pixelated, but it's still enough to recognize that he was wearing a black suit and a white shirt. And once we go to the third zone, like all the information is lost, everything is just gray blur. Now the same concept of multiscaling can be adapted to a protein. It's unusual. I mean, it's very rare that the entire protein is involved in a reaction, or let's say in absorption. Usually it's an active site or a chromophore, which is much smaller. So we can use this smaller size region and describe it using quantum chemistry. And then when we go outside from this active region, we can describe the remaining part, the protein, at a lower level of resolution. So similar to here, we reduce the resolution and we go from a nucleus and electron picture to a pure nucleus picture where the electrons are represented in empirical force field parameters. We can also achieve this third zone by just simply using a dielectric constant, and then we lose all the atomistic details. So now the first question, when we look at the cyanobacteriochrome, this is the protein, this is an insight into the chromophore and the binding site. So the first question is, how do we divide the QM and the MM region? And so since this is a relatively new protein, we decided to test two different regions. One is composing 66 atoms. It contains these 4 rings of the pyrrole. And the other one contains also the 2 propionates, which are not conjugated, and the cysteine, as well as the histidine residue and the aspartate. So this region is composed of 106 atoms. Now, when we want to calculate the spectrum, typically in quantum chemistry, if an isolated system, a molecule or a small chromophore is treated, one usually locates the minimum on the potential energy surface and then computes for the same geometry the excited state energy. However, in the protein, the situation is dramatically different. The potential energy surface is very complex, and there are a lot of minima which are actually quite close in energy. So in order to sample this surface, this is much more complicated due to the higher number of degrees of freedom. We have okay, so for some reason the movie is not working, but like what it actually is showing is that, I mean, we're doing molecular dynamics and then everything is supposed to move here now. And yeah, okay, it doesn't work, but you just, I mean, can imagine how everything is shaking and moving while we do MD. And then from this molecular dynamics simulation, we take snapshots. And so I would just, before I show you the spectrum, I would just like to summarize. So we have a 1 nanosecond QM-MM dynamics that we use for sampling. Here, the QM region is treated using the semi-empirical DFTB method. And then we use both QM regions for excited state calculation. We take 100 snapshots every 10 picoseconds. And we use ADC2 with a double zeta basis set for calculating 10 states for the small QM region and 15 states for the larger QM region. Okay, and then the interaction is between the protein and the chromophore is treated at the using electrostatic embedding. So, so this is how a computed spectrum looks like. We have sticks, 100 sticks. The black sticks represent the excitation to S1, red S2, and so on. So here you see the 10 states for the small QM region, and then we put a Gaussian on top of each stick, and we add them up. And then we obtain a spectrum which looks similar to an experimental one. So now if we do it for the red and for the green form, and we compare them, indeed we are able to represent, indeed we are able to obtain a green shift. So it looks like for the small QM region, which is represented by a solid line, and the large QM region represented by the dashed line, the spectral shift is almost the same. So it's a quarter of an electron volt. The experimental shift is something like 0.32. So it's not too far off. Now, the first thing, so first we established that actually the smaller QM region is enough to reproduce the spectrum and the shift. Now we can do something which cannot be done experimentally. We can just simply remove the protein environment. So we can set the point charges to be 0, and we can recalculate the spectrum. So as you see, as a consequence, the spectrum has shifted for the red and for the green, but the spectral shift between them has changed just by 0.04 electron volts. And that's why we can make the first conclusion from this simulation that actually the hydration, oh, sorry, I forgot to mention, we also set the charges of the water molecules to be 0. So the hydration mechanism is not valid because removing the protein and removing the water charges doesn't affect the spectral shift. It doesn't go to the far right, it stays in the green. So we ruled out this mechanism, and now the only remaining one is the trap twist. So in order to analyze the trap twist, we have looked at the twist between the A and B ring, this is the x-axis, and the C and D ring, this is the y-axis, okay? So here you see 100 snapshots from both red and green form, and you see that they form nice clusters. There is one cluster in this region, and the star is actually the crystal structure, the values for the crystal structure, and you see a nice cluster for the green structures, and the color of the points represents the absorption wavelengths. So it looks like they give nice clusters. Now, if we compute the Pearson correlation coefficient, which is supposed to be 1 if there is a very strong correlation and 0 if there is no correlation, we get actually quite low values for the A and B ring twist. In fact, if you plot the twist of the A and B ring versus the excitation energies, the correlation coefficient is close to 0, so I don't show the values. But for the C and D ring, the values are actually 0.1. The correlation coefficient is 0.1 for the red color and 0.4 for the green. So it's not like you're really convincing a great correlation. So we went a step further, and then we applied wave function analysis. So in this wave function analysis, a special focus is laid on information encoded in the transition density matrix and its phenomenological analysis in terms of the electron hole picture of the excitation. So for this purpose, we have taken the chromophore and we divide it in 3 pieces. One is containing the A ring, the second one, the B and C ring, and the third one, the D ring. And then we got this electron hole correlation plot. So here is the location of the hole, and here is the location of the electron. And in this example, if the hole and the electron are located on the BC unit, then in this correlation plot, we'll have an entry of 1 in the middle because it's on BC. If it's a charge transfer excitation from BC to D, then we have an off-diagonal element here being 1. And so if you do it for all 100 snapshots and add the values together, we get the following electron hole correlation plots. And then we look at the secondary off-diagonal elements. So these values describe the charge transfer. So if we add up all these values here together and we sum them up for the red form and the green form, which basically describe the character of the charge transfer or the charge transfer character, then we get much better correlation coefficients. Then for the red form, we get 0.5, and for the green form, 0.8. So while if you compare only the twist between the C and D ring, the correlation coefficient is much smaller. So what does it mean? It means that it also, this wave function analysis takes into account other geometrical features, for example, bending, bond stretching, bond elongation, and other distortions, which hinder the conjugation. And so it's not only the CD ring, it's not only the twist between C and D ring, but it's also other deformations in the green form, which make a difference and cause the green shift. So the take-home message for the first part is that it's rather the trap twist mechanism than the hydration mechanism, okay? And then we also conclude that the charge transfer number, or the effective conjugation length, in other words, is responsible for the red-green spectral tuning in this specific cyanobacteriochrome. So we also establish a simulation protocol, how we get a spectrum from 100 snapshots, and we use it also for the second part of my talk. So now, in the second part, we want to address something called the heterogeneity, which has been reported for this specific family of cyanobacteriochromes in a range of experimental studies. So first I want to mention what do I mean by heterogeneity. There are different forms of heterogeneity. So one could be, for example, the structural heterogeneity. So there could be, you see the chromophore is quite flexible. There could be different conformation. There could be different protonation states. And also in the protein environment, the side chains of the amino acids can also rotate. One important conformational change is that the D ring here can be above the plane spanned by the BC unit, or it can be below. If it's above, it's called the alpha substate. If it's below, it's called the beta substate. I will come back to this form of conformational heterogeneity in the next slides. But then we can also have spectral heterogeneity. So we can have different peaks of wavelengths. We can also have different fluorescence properties. And then we can also photochemical heterogeneity. So for example, excited state lifetime can vary, quantum yields can be different. Here is an example from Lagaria's lab together with Delma Larsen. So here they studied one cyanobacteriochrome and they measured 3 different excited state lifetimes for 5 picoseconds, 50 and 350. And all of them have different photoproducts and different yields. And last but not least, there's also the biological heterogeneity, because depending on the reaction, then the products can have different biological activity, and some of them can induce a signal to the cell, some of them don't. So now, we stay with this red-green family of cyanobacteriochromes, and we look at a recent paper which combined MD simulations and NMR. And they claim that the chromophore is heterogeneous. So what it means is that in this MD plot, you see the angle between the C and D ring, which is here, it's changing from -40 to +40. So it means the ring is going above the plane, then below the plane, and then back. Okay, so they have further substantiated this MD simulation using NMR chemical shifts. And before that, actually, a resonance Raman study with a different MD simulation of the same protein has concluded that the chromophore is homogeneous, so there is no heterogeneity. This was then further supported by NMR and time-resolved absorption and fluorescence of a related protein. So the question is like who is right, who is wrong? Is there a heterogeneity or in the chromophore, yes or no? So in order to look for it, this was actually the work from Aditya, who is a PhD student in my group. We set up the simulation for this protein. And then the first question that arose was, what is the protonation state of this amino acid highlighted in orange? This is the histidine with the number 322. It can be either doubly protonated or singly protonated. So when Adi came with this question to me, I also didn't know the answer. And I said, okay, let's try both. Let's try the single protonation and the double protonation. So he carried out molecular dynamics simulation. And then what we found is here you see the RMSD, that in case of the doubly protonated histidine, which is the DPH model, there was an increase in the RMSD. In case of the singly protonated, there was no sudden increase. So if you looked at the RMSD if you look only on the CD dihedral angle, we saw that actually this increase is due to the change from alpha to beta substrate. It means the D ring was going from up, so from being above the molecular plane to below and then back. So, but this was only one trajectory of 1000 nanoseconds. So the question was like, is this reproducible? What happens if you run more trajectories? Maybe also the singly protonated histidine shows the same behavior. So Adi repeated this for 2 trajectories, and then this flip in the D ring was not observed. So we decided to do some sampling. And I here have one slide to explain what I mean. In usual molecular dynamics simulations, there is an unbiased sampling. So it means usual molecular dynamics, we have to run a very long simulation and average forces from all the conformations to obtain the potential of mean force, which is related to the free energy and which will give us the final answer. Now, this can take a lot of time, and for protein in solvent, and you can never be sure that you really sampled enough. However, then you can turn to, instead you can turn to biasing on an artificial potential sampling. One of them is so-called umbrella potential. And then you can introduce restraints on the potential energies along a specific reaction coordinate. So in our case, we know that the CD ring is twisting. This is our reaction coordinate. So in a very simple picture, there's like one minimum of one twist, let's say alpha, and there's another minimum beta. So what we do is now we put these constraints, the red constraints, they look like closed umbrellas, and that's why it's called umbrella sampling. And the only requirement is that these umbrellas are overlapping. And then we sample each region selectively by using this constraint. And then from there, we reconstruct the potential of mean force. So after doing this umbrella sampling, what we have obtained is the following. In case of the doubly protonated histidine, and indeed there are two minima for the alpha and the beta substrate. And these two minima are separated by a very small barrier. However, if we look at the single protonated histidine, there is only one minimum at the alpha subset, and the minimum for the beta subset is very shallow. So basically, if you reach it, you immediately convert back to alpha. Okay, so I guess like if there are experimentalists here in the audience, I mean, the question is, how can you prove it? What's the experimental, how can you check it? So then we went on and we computed the absorption spectrum using the same protocol I showed you before. And we obtained for the doubly protonated histidine, we have for the alpha and the beta subset, the absorption spectra look actually identical. I mean, the shift is too small to be observed experimentally because the bands are very broad. And now for the singly protonated, the absorption maximum was in the same region. So we couldn't distinguish it by using conventional linear absorption. So then we turned to circular dichroism, which was already introduced by the previous speaker, by Professor Kenneth Root. And it turns out that this the movement of the D-ring above and below the plane changes the helicity of this chromophore. And therefore, if you look at the beta substate, it has a positive rotatory strength in the CD spectrum for the main band, while the alpha subset has negative. Now, also the ratio between the main band, which is the Q band, and the minor band, which is the Soret band, is different for alpha and for beta. So if you have a presence of both of them in the solution, as we have shown with umbrella sampling, you will probably have some kind of compensation. Now, in the single protonated histidine, there is only one single subset. This is the alpha subset, and it has a negative rotatory strength. So then we went through the literature and we looked at related proteins, and we found that indeed both cases were reported. So here is the experimental spectrum. So the solid line is for the red form, which we studied. The dashed line is for the green form, which we have not studied yet. And you see, indeed, there is a positive rotatory strength or positive band for the Q band and the positive for the Soret band. And this can only be explained by the presence of both alpha and beta subsets at the same time, because they have different ratio in the bands. Probably there is I mean, from our estimation, there is 3 times more of the alpha subset and beta subset, and that's why the band here is like very small positive. And since the ratio between the Soret band is changing, and you have more of this alpha subset, we have a positive signal here. And then there is another protein which is not heterogeneous like this singly protonated histidine. And indeed, for the red state that we studied, we have here a negative band for the Q transition, and we have a positive one for the storage transition. Okay, so the second thing, or the take-home message for the second part, is that it turns out that the protonation state of this histidine is the key in order to have structural heterogeneity. And we actually we think that the contradiction between the two experiments that I showed you before is due to the slightly different modification of the protein and also at different pH values where the experiment was conducted. And so we have, from our simulations, we have found that UVV spectroscopy cannot distinguish between alpha and beta substates, but using circular dichroism allows you to distinguish between them. Okay, and that's the end of my talk. I would like to acknowledge my group, and in particular, Chris